Hello and welcome to our Lakeside chat. We are David Wofford and Katie Noonan, co-chairs of the Rotary Nature Center Friends. And we're so happy that you can join us tonight.
As we begin our program, it is important for us to acknowledge that we are on stolen land, that Lake Merritt is part of Ohlone territory. We hear now from Corinna Gould, spokesperson for the Confederated Villages of LaShawn and co-founder of Segurate Land Trust.
Good afternoon, relatives. My name is Corinna Gould. I am the spokesperson for the Confederated Villages of LaShawn. We are here today at what most folks think of as Lake Merritt. We are in the territory of Huichin. Huichin is actually a territory that encompasses 6 Bay Area cities: Oakland, Berkeley, Alameda, Emeryville, Albany, and Piedmont. And so this is a place that my ancestors have been since the beginning of time. And this was a place of abundance. I'm so happy that people from all walks of life that now come into our territory can enjoy this beautiful place that my ancestors have enjoyed since the beginning of time. My relationship to the land that I have been born to, raising my children and grandchildren here, has been to tell the story, the truth of what happened on this land before other people came here. I'm hoping that as we begin to learn these lessons of fires in California, the pandemic that's happening, that human beings come back to living in reciprocity with the earth.
Thank you, Corinna, for a powerful message that we need to hear today as well. I want to thank everybody who's here tonight, and we're looking forward to a wonderful program with Rebecca. As we start our show, I would like to share a few things. A reminder to everyone, we are Rotary Nature Center Friends, an independent, all-volunteer 501c3 nonprofit since 2021. The focus of our activities is community science and environmental justice. We involve people from all throughout the community in learning about the ecosystem that we all inhabit and how we can build an intergenerational local community serving all of the people of Oakland. I want to thank at the very beginning our supporters and people we've done some amazing things with in the last month. We hosted Save the Bay for a visit to the shoreline. We've been working with Alameda County-wide Clean Water Program to provide creek exploration and study activities with local high school and others to explore one of the tributaries to Lake Merritt. And we're just so happy to be able to interact with all of these wonderful people who care about Lake Merritt. We are starting tomorrow a Urban Estuaries California Naturalist Certification Program. Really looking forward to having an even broader and tighter community of people who are interested in learning more and communicating to others the importance of where we live and our relationship to the people, the land, and the ecosystem. I would like to add at this moment that we are my co-chair David Wofford is not able to join us tonight because he is ill. However, he is getting better, and he wanted to share with you how much he wished he could be here and how much he is looking forward to being involved in our activities in the coming month, including the California Naturalist Program. We'd also like to mention another of our wonderful contributors, Jim Covell, who is also recovering from illness tonight, but we look forward to his joining us as soon as possible. So, at this point, I think it is time for me to introduce our speaker, who you've already met if you were in the trivia program, Rebecca Abrams. So, Rebecca is a lifelong lover of museums and nature. She earned her BA in biology and Masters of Education from UC Santa Barbara, and then a Master's of Museum Studies from San Francisco State. She has 15 years of experience working in natural history museums and is currently the collection manager of fishes at the Museum of Vertebrate Zoology at UC Berkeley. And she is a board member of Rotary Nature Center Friends. We're so lucky to have you with us tonight.
Welcome to the Fishes of Lake Merritt. Like Katie said, my name is Rebecca Abrams, and I'm the collection manager of fishes at the UC Berkeley Museum of Vertebrate Zoology. Please feel free to email me any questions, comments, concerns, fish facts, I don't know. My email will be here and it'll be again at the end. I would love to talk to you about fish. And there we go. Okay, that would be awesome. So just as a quick overview of what we're going to do for this evening. And once again, if you have like a burning question, put it in the chat and Janai will unmute you and let you talk. Otherwise, she'll collect them and we'll talk them out at the end. Here's what we're going to do. We're going to go through some basics, then a brief history of Lake Merritt, because you can't understand the fishes in the water until you know where the water comes from. We'll look at some historical fish data, because this is not just about the fishes that are there now, but the fishes that have been there in the past. And we're also very interested, particularly at natural history museums, as repositories of basically time and of history. That's, natural history. We want to see those changes through time. And so we will be looking at as much historical data as we can. We'll be looking at our current work and then also what's going to be happening into the future. As another brief note, I'm going to be referring to the body of water where this is all happening as Lake Merritt. I know it's not a lake. It's a tidal slough or a tidal lagoon, but we're going to call it Lake Merritt, just for simplicity. Oh, clicky the button. Okay, so some basics. What's a fish? Well, I'm going to be using the colloquial term fish, which covers everything inside one of these bright blue boxes on this phylogenetic tree. And that's fine. If any of these animals showed up at the Museum of Vertebrate Zoology, they would be shuffled off into my collection because they live in the water and have fins, kind of. The problem is evolutionarily and taxonomically, when you start to organize things by how they're actually related to each other on basically a DNA level, which is how we do this nowadays, you very rapidly come to the conclusion that the term fish really doesn't exist. And there's even a book called Fish Don't Exist. And if we were to actually call things fish properly, and to make it what we would call monophyletic rather than paraphyletic. So monophyletic meaning it only has one common ancestor rather than a whole bunch of common ancestors all over the place, which is what it has now. We would have to go back to where this blue dot is on the family tree, on the phylogeny. And that means that things such as this lovely rooster and mammals would also be fish. And if you talk to most ichthyologists, they're thrilled about this. And if you talk to most mammalogists and herpetologists, they are not thrilled about this. So for the purposes of our talk, we're going to be going with colloquial fish. If you would like to get into a debate about this, please go to the American Society of Ichthyologists and Herpetologists conference and ask, what is a fish? It's riotous and quite fun. Some of the most common questions that we get asked, we as in Rotary Nature Center friends get asked when we're tabling down at Lake Merritt, are there's fish in there? Mostly said incredulously and confusedly. Isn't the water super polluted and gross? And wait, it's saltwater? Again, incredulity, confusion. And so the title of the talk, The Fishes of Lake Merritt, should basically answer this first question for you of yes, there are fish in Lake Merritt. There are many species of fish in Lake Merritt, and they're doing pretty darn well. Whether or not all those fish species are native is a different question and a different comment. But there is enormous amount of fish diversity in Lake Merritt. And to the question of, isn't it polluted? We will get there, and it's saltwater. Yes, it is saltwater, and the history of Lake Merritt will tell us why it's saltwater. So if we look at this image, this is just a Google Earth image. We can see Lake Merritt. It's the kind of Y-shaped, or I don't know, pointy V, U-shaped thing in the kind of upper right quadrant of the image. And then we can see the San Antonio Creek channel coming out of it, and then the channel between Oakland and Alameda. So this is our modern view of Lake Merritt, and you're going to see this again and again and again, and you're going to see it change. And so one of the big things about understanding natural history is understanding where we were, or where a place was, and where it is now. And you have to understand the geology and the hydrology in order to understand what lives there now. That's the basis for ecology. You can't understand what plants and animals live there without understanding the rocks and the water sources that are the foundation of that. So Lake Merritt, and a huge thank you to Andrew Alden for his incredible geologic work and histories of Lake Merritt that I drew on heavily for the background information for this. Historically, Lake Merritt has three sources of water: freshwater streams, the Glen Echo drainage, what he terms the Merritt Creek drainage, and then freshwater, so rainfall, sorry, and then the kind of southern inflow, which would be the saltwater tidal slough, which comes in through what it is now termed San Antonio Creek. Lake Merritt has been a source of natural resources for thousands of years by the Ohlone peoples, and was used by them and is still used by them, and to this day, though under very different circumstances. By the time the area was under ownership management by the Peraltas, by the Peralta family, the Ohlone people's lives had been radically changed, and their usage of the land had also been heavily changed. After California's statehood in 1850, when the Peraltas no longer owned the land, Samuel Merritt in 1854 bought 23 acres of land around the shoreline of what was then a tidal lagoon. This map is from 1857, and you can see what was Lake Merritt as a much more open waterway to that channel between Oakland and Alameda. And at this point, that channel is roughly a quarter mile wide, so 1,300 feet. Now, as we'll see later, that channel is significantly restricted. And so the flow of water in and out of what was then really a tidal slough could be quite extreme and controlled by the tides. Um, buying property around what he thought was a beautiful lake, and then at low tide when all the water goes out, it's gonna smell because it's a tidal slough and the algae and things start to dry out and it gets a little stinky. So he wasn't jazzed about his beautiful waterfront property starting to smell. So in 1869, he used his very own money to build a dam across the mouth of the slough near where 12th Street is today. And that dam was up for well over 100 years. This started the culverting in, the control of Lake Merritt that we will continue to see sort of to this day. Some reversal has started now. This map is from 1872 and shows the start of the land tract sales around Lake Merritt. 1870 is when we start to see the big changes. This is the first time, at least that I could find records of the lake being dredged, or what is now called the lake. Keep in mind that as a tidal slough, Lake Merritt is shallow, maybe 6 feet deep. Today, at its deepest, it's 10 feet, maybe 15 in like really deep spots, but you could like walk across most of it. The surrounding marshlands are starting to be filled in because, again, marshlands are considered stinky, unsightly, and also excellent spots for housing. And also, this is when San Antonio Creek is culverted from a quarter mile wide, 1,300 feet, down to about 100 feet wide, which is roughly its current width. This is also the fabled year when Lake Merritt Wildlife Refuge was founded. But keep in mind, it was founded as a hunting preserve. And this is the mindset of the time, which had great knock-on effects. We preserved the area for bird populations, which, thanks to the great desire for hats and feathers and all sorts of other things, birds were definitely imperiled, but we're protecting this land in order to later exploit it, which is the same thing that we set up national forests and national parks for. So it's kind of an interesting mindset for us to wrap our heads around, I think, looking back on. This is an interesting map from 1885 that proposes a boulevard around Lake Merritt, which we have today. So I thought this was kind of neat. This photograph is from 1908, looking south toward that 12th Street Dam at San Antonio Creek, the little blue stars right above it. You can see the urbanization around it. You can kind of see power lines and what I'm assuming are probably telephone lines. And you can also see this doesn't look very, I don't know, this looks quite urbanized and not particularly wild anymore. You can also see some very large houses around the edge. Keep in mind that there has been a true population explosion in San Francisco, starting in 1849 with the gold rush. California became a state in 1850 because of this. So between 1848 and 1849, the population went from about 1,000 people in San Francisco to 25,000. That's just astounding. Between when Samuel Merritt bought his property which was the population in San Francisco around that time was give or take 35,000, to when the wildlife refuge was established in 1870, the population was up to 150,000 people, and it's just going to keep increasing. This is a highly dense urbanized area. And also, with this urbanization and all these people, they're coming on ships. They're not coming across on a railway yet. Not really. And so we can say with pretty decent confidence that this is where the start of our delightful aquatic invasive species come from. They're probably coming in bilge water and ballast stones from all these ships from the East Coast and Asia and other ports of call, but those are our main ones. We also start seeing dredging for deeper harbors. And so this leads to profound ecosystem change that we are going to see for the rest of the history of the Bay Area. 1906, we see the Great San Francisco Fire and Earthquake, and we see again another huge urbanization event happening in the East Bay as people flee across the bay. Interestingly, around the shores of Lake Merritt, and this was something that I stumbled across and kind of had to put in, a Chinese refugee camp was established called the Willows. This is a photograph of it from the time, and then also an artist painting that I believe was done contemporaneously. And I'm not sure if this was set up because the land was considered fairly undesirable, considering this 1908 newspaper that said that the lands around it were a, quote, marshy eyesore in place of reeking odors. Unclear. Or if that's just where people ended up. But also, with more and more people came pressure on the lake and on it as a resource. This was still a time where dumping waste out of sight, out of mind. Let's be real, we still kind of do this. So in 1908, the city of Oakland decided that it would start filling in land between 8th and 12th Streets, down at the bottom, the southern end of the lake, with mud dredged from the lake to turn it into a 30-acre garden spot the city would be proud of. So again, more dredging, more modifications to the lake. Today, Lake Merritt is heavily modified from its original kind of wild source. This doesn't mean that it's bad, it's just different. It is fully enclosed in concrete walls. There are 62 drains that flow into it from the city. So we get lots of storm drain runoff. We have rainwater runoff. We have runoff from lawns, from streets. So we're getting fertilizer, we're getting oil, we're getting dog waste and human waste are huge sources of contaminants for any urban waterway. There is a tide gate that does alter the flow of salt water in and out, that is opened sometimes. Fish can move through that. It's got pretty large gaps in it, so fish as large as bat rays can move in and out of it. Both of the major inflow creeks are now restricted or fully culverted over. Glen Echo Creek is still visible, and there's restoration happening along it right now. The algae that a lot of people like Samuel Merritt found so offensive is removed yearly to up to a depth of 6 feet. We will talk about this later. There are 2 small aeration fountains installed within the lake. These are another issue that we will not be getting into today. And in 2010, the 12th Street Dam was removed, and this did lead to possibly more mixing of the waters in the lake. So what do we know about the fish in our lake now that we've kind of oriented ourselves to this human modified body of water? Well, we have a lot of records of fish kills or fish die-offs. These are from the Oakland Tribune, starting in 1939. And they suggest maybe more frequent saltwater flushing would help. 1949, the city lets in saltwater due to what they say is pollution. 1951, the city cites low dissolved oxygen levels and water temperatures. 1967, the city blames a windstorm. That's a good one, I don't know. In 1974, they say hot weather. So before we had low water temperatures, now we have hot water temperatures. Unclear. Keep in mind that between 1939 and 1974, it's unclear as to if these hypotheses were really tested with water quality testing. I do love, in particular, this image of who's this man's name? I've missed his name. Yes, just shoveling carcasses of striped bass out of the lake into a wheelbarrow. 2016, we know that there was a large bat ray die-off, most likely from a parasite. And then, of course, the 2020 algal bloom that had so much coverage. And we do have samples of these fish. And we can say pretty conclusively that, yes, it was an algal bloom. And because we have samples and we have extensive photographic evidence, we can start to test these hypotheses and gather modern data on them. We also have some really interesting data from smelt derbies. And so starting in 1939 and all the way through 1966, possibly later, again, if you have records of these smelt derbies, including personal photos, I would love these. So it started out with the city hosting these, possibly in conjunction with a private fishing outfit along the lake, and then seems to have been taken over by the local Lions Club. But I'm not entirely sure. I tried contacting them, didn't quite get back to me. But these were fishing derbies for children, and they were fishing for either or top smelt silver sides and the other kind of top smelt. Brain is blanking. I'd like to thank Jim Cavell for providing this lovely color photo from 1961. And the basic thing was you caught as many fish as you could in roughly 6 hours. This says to me that there were a lot of top smelt in the lake. We don't see these huge numbers now. And so the question is, what's going on? So as we go forward, keep an eye on those smelt populations, those top smelt populations. Those interesting things are going to change. I got into this whole Lake Merritt rabbit hole because we have these beautiful specimens at the Museum of Vertebrate Zoology from 1963, collected by John D. McCarty Jr., who was the assistant to James Covell's dad, who was the first park ranger is the wrong word, who ran the Naturalist Center at Lake Merritt. And he collected all of these on October 30th, 1963, and we are fortunate enough to have them preserved. So this is a snapshot in time. And if you would like access to them or would like to see their records, they are available online. They're very, very cool. You can also come visit them if you schedule an appointment. So here's what some of them look like. You can see we have, let's see, these are the prickly sculpin and the staghorn sculpin, our three-spine stickleback, our shiner perch, and our arrow gobies. The other ones, the photographs weren't as pretty. But this is really what got me started on this is we have these local specimens and no follow-up. So we can do a time series, but there was nothing to compare it to. And then it turns out that the California Academy of Sciences, here on out known as Cal Academy, also has specimens, but theirs are a little bit more all over the place. They've got one top smelt from 1928. They also have a 1963, I also think from John McCarty. They've got a 1966, a 1967, and then this weirdo that I really have to go look at, 1995, a Pacific fat sleeper who's this little chunky friend down at the bottom. They are not supposed to be farther north than LA County. So I think that this guy's stowed away in Bilgewater because it is hundreds of miles north of where it should be. I don't know. I have to go look. But this is roughly, this is all the scientific specimens that I have been able to track down so far. I have been told that there may be specimens at Laney College and at Merritt College. I have been working to try to find those. I was told that there may have been some at the Oakland Museum of California, but those seem to no longer exist, or they are buried in the collection somewhere. So again, if anybody has contacts, I would love to be able to find those specimens. Disassociated specimens from their data are the bane of a collection manager's existence, and we would love to find them again. So moving forward in time, we have this series of observational data from Katie Noonan and the other teachers that she worked with and her students from the early and mid 2000s. And so we can see just from the kind of common fish list that she had her students watching out for, that these are things like Pacific herring and shiner perch, which in our collections now are not common. This is kind of interesting. And then on this data sheet on the right, you can see dozens of three-spine stickleback. Oh, Katie, is this an emergency? Go for it.
A little emergency, not really. No,
Yeah.
the fish chart on the left is actually Jim Carlton's fish
That's
chart.
right.
Yeah, but the one on the right was from our Environmental Science Academy work in the 2000s. Yeah.
Yeah, that's right. Yeah. So, and while that collection of sticklebacks is from across 4 days of collecting, that is tenfold the number that we have found in a single day or across 4 days. So interesting to see possibly a shift in the ichthyofauna again. And then we can also use what we would consider community science observations from platforms like iNaturalist to look at trends over time. And we know that this is going to skew toward charismatic species, things like striped bass and bat rays that are big and easy to identify. And bat rays are just, let's be honest, they're really cute. So people are going to take pictures of them. But we also may see an increasing number of invasive species compared to the early 2000s data. This is something to dive into. We also know that when we look at data like this, there are issues with identification. And so if it's kind of a small silvery fish, it's going to take more work to ID it than it will something like a striped bass or a bat ray. We also see a high number of fish like Chinook salmon observations. Again, it's effectively what's known as charismatic megafauna. They're big, they're literally shiny, they're very cool. So they get more pictures. This does not mean there have been 99 Chinook salmon in Lake Merritt in the same time period that there has been, 2 three-spine sticklebacks. Absolutely not. The Chinook salmon are just a lot easier to photograph because, they're 1.5 feet long compared to something that's 2 inches long. So something to keep in mind. The other thing that we would like to be doing, we meaning the powers that be at the fish group, is to add data from predation photos. And these are some of the best fish photos, which are things eating fish. There are a fantastic set of these of a Chinook salmon being eaten by gulls. This is perfectly valid fish observation data because it's still a fish, it's just not alive anymore. And so this is a known method for adding data from photographers. So we basically have to trawl through the bird data, the bird photographs, and look for birds with fish. And then we can add more to our data set. So some questions from our historic fish data that are brought up are, where did all the smelt go? Where did all the sticklebacks go? And then is there a turnover in invasive goby species from yellowfin gobies to chameleon gobies? And we're starting to see this a little bit more in our current work. And is that a true turnover? Is it seasonal? What's going on? Again, more data, more better. So what's going on now? What are we doing right now out at the lake? And would you like to join us? So I would like to give a huge thank you to Katie Noonan for getting me into all this. Actually, to Lila for stopping to talk to me about fish when I was just staring down at them one day and saying, "Hey, you should talk to Katie." And so Katie does this work so much, and the Rotary Nature Center friends, and then all of their volunteers, and Janai with her incredible plankton work. And really, the amount of data that they and the students and all their other partners are able to collect is astounding and truly invaluable. And the time span that this data has covered is amazing. So here you can see two of the Achieve Academy students doing a plankton tow. You can see some of the plankton under the microscope, and this is water quality data from the SOND device that the students also know how to use. And then this is some of the salmon. These are the Chinook salmon. They're hatchery fish for the most part. Ooh. Yes, Katie, yes.
We don't know.
That's
'Cause,
part of what is,
yeah.
that's part of what is to be found out.
Unless they have a coated wire tag in their noses,
Yeah.
we don't know.
So that's part of the interesting thing, is that salmon find their way back to their spawning grounds by smell. These fish are, if they are hatchery fish, they were not released down Glen Echo Creek. So why are they coming back into Lake Merritt to presumably swim up Glen Echo Creek? Why are they a little bit lost? So this is part of the ongoing work. And these things that kind of look like, I don't know, rice grains or oatmeal. These are otoliths. These are the ear bones of the salmon. Yes, they have ears and ear bones. Well, they don't really have ears, but they have ear bones. And this is a way to tell their age, as well as do an enormous amount of chemical analyses on them. Also, keep in mind that by the time salmon come back into freshwater to breed, they're going to die. They have gone from their saltwater, they've gone from their freshwater, basically their freshwater set of DNA, to their saltwater set of DNA, and they can't switch back when they hit freshwater. So yes, these fish look beaten up because they're dead. Their death is inevitable because they're coming back to breed. I feel like that kind of needs to be said. Also because Fat Bear Week just ended, and that is the epitome of salmon runs up in Katmai, up in Alaska. So many salmon. Those fish are going to die anyway. So salmon are absolutely fascinating. Fascinating fish, and there's a ton of research on them, and so this adds more to it. So this is the work that I've been doing with some undergraduates at UC Berkeley, and with Katie and with the Achieve scholars, and really anybody who's around to help, including recently some cadets from the California Highway Patrol came out the other week for Crete Beta. It was great. So what we have are these traps. They're minnow traps. You can see a beautifully clean one here. We let ours get colonized by all kinds of friends off of the docks, what we call dock fouling organisms. And we see who comes to visit. And we have these mostly near the boathouse at Lake Merritt. And we find all kinds of friends. And it's pretty neat. So on this page, you can see a bay pipefish, which is native. you can see a rainwater keely, which is not, a yellowfin goby, which is not, and this is a little staghorn sculpin, the vertical picture, which is also native. And then this is just one of the data sheets for a particular day, just to show you what it looks like. We dump them into a little bucket and we record them. And then this is what our current collections look like when they come back to us. For the most part, my kind of rule of thumb as a collection manager, things I have to think about are, do I have space? And that answer is rapidly becoming no, which is interesting. And also the scientific value of the organisms and also their role in the ecosystem. So when I go out and collect, I am now not collecting pipefish. Pipefish are native, they are also a predator. So this particular jar came were pipefish that had died already. So that is why they were kept. But I am currently only keeping non-native species. Native species will be photographed and recorded as observation only because I just, in my heart of hearts, I can't take the native species unless they're already dead. One of the other interesting things that happened over the summer, basically through serendipity, was this algae harvester that the city contracts with every year. And when they do this harvesting or cleaning is arbitrary, according to the people that were doing it. They're a company that contracts with cities up and down the West Coast. And basically, it uses a conveyor belt to collect algae down to 6 feet in depth. Keep in mind that Lake Merritt is often shallower than 6 feet, so they are scraping the bottom and scraping up everything. And so they were kind enough and didn't look at us too strangely when we asked to basically dig through their piles of algae that they dumped onto a huge flatbed. And so these are buckets of rainwater keelies that we found alive in the algae and yellowfin goby that were also alive in the algae. They were humanely euthanized and put into our collections. And these are the fish that were found dead in the algae harvester. You notice all the pipefish. Interestingly, we have some arrow gobies also, which are native. So this was quite a serendipity, but also neat because it gave us access to parts of the lake that we don't normally get to or haven't gotten to yet, meaning the far side of the lake from where the docks are that we're at, and also the middle of the lake. So some questions that it just brought up are, what effects is the algae harvester having on fish diversity? Does this actually help with harmful algae? Is this changing the dissolved oxygen content, which is a big topic when one is at Lake Merritt? Were any environmental impact studies done beforehand on the effects of using this algae harvesting method, and will they be? And how do we monitor the proportion of native and non-native fish killed? Simply just extremely limited data set. Only the non-native fish survived. Meaning if we go back, the rainwater keelies and yellowfin gobies, and also the chameleon gobies, which I forgot to take a picture of that day. But our native fish, which are basically the entire right-hand side of this picture, did not survive. And pipefish are extremely fragile. You kind of look at them wrong and they die. But that was quite interesting. Keep in mind that the algae harvester basically just rolls the algae. And this is kind of death by blunt force trauma. This is not a fun way to go. Interesting to me that the arrow gobies did not make it, because gobies can usually handle a bit more, can handle lower oxygen levels and kind of being out of water more, but maybe arrow gobies are just a little bit more fragile. I don't know. So I think this is a very interesting data point that is unfortunately not easy to replicate because they're not there the same time every year. They're just there whenever they have time in their schedule. So we can't even predict when they'll be there next. Yeah, it's kind of an interesting thing to get to see. This is our work seine netting. We haven't really gotten to do this more than once. But a seine net is essentially a rectangular net between two poles. It has floats on the top and weights on the bottom. And you walk it out into a big loop, and then you lift it up, bring it into shore. And what's nice about this is it allows us to go for really, really tiny fish. And so this is over at the beach by the bandstand. And so hopefully with the power of undergraduate helpers, undergraduate research, like Haylin here, who's standing with the hat on and pants on in the water. She's one of my fantastic undergraduates. We'll be doing a bit more of this. So what's the future of fish research looking like at Lake Merritt, at least from my perspective? I am merely one person and some undergraduates. More field data, more better. Katie and I have been setting more traps, and we're trying to set traps mid-lake, not just at what is on here labeled as Adams Point, which is basically where the boathouse docks are. There are other docks. There's the boat club docks. There's a little dock at 12th Street, but it would be awesome to see what's in the middle of the lake. Cast netting, which is that kind of thrown circular net, that would be really cool to do. Step one would be learning how to do that. But I figure I can practice on a lawn before I have to do it in a lake and fall out of a boat doing it. And then more seine netting also. You can seine from a boat, and you can also seine from the water. And again, luckily, Lake Merritt is just not that deep. So you can kind of walk in and do it. And then this is one of a new type of trap that I'd like to try out. They're called eel traps. They're very small, not much bigger around than maybe I don't know, 2-liter bottle. So maybe they can sit down in some shallower areas. Also getting traps and water quality data in the inflow creeks. Glen Echo Creek, just over just over Grand Avenue, and then also farther up Glen Echo Creek would also be really great. And then maybe down into the channel, down into San Antonio Creek. We've tried it once before, but the current there is very strong and the traps are vanished. So we'll work on that. But there are some neat fish down there. And then analyzing more of that historic fish data. So with that, we can begin to analyze long-term trends in ichthyofauna data. Looking at pictures and doing things like literally counting the number of fish in that picture, you can actually begin to estimate fish populations, particularly from fish die-offs. I can see from this photo that there are multiple species of fish in here. Some of these are anchovies, some of these are topsmelt, and some of these are different species of goby. Yes, they all look like little dead silvery fish, but they're not. And there's a lot of pictures of this. This is the 2022 fish kill in the news and also on iNaturalist. And that is a huge amount of data. So that is, I hate to say this, a great job for an undergraduate. And we actually have them do this a lot. And then we can use that data to see what was there. If you have memories, photos, written records about Lake Merritt's fish, if you have a picture of yourself as a child holding up a fish you caught, I would love to have that photo. We have an archival division at the Museum of Vertebrate Zoology. We have archives, but they can also be entered as fish observational records. And it just adds to the body of knowledge about this incredible place that we live near and that we interact with, for some of us, every day. And the more information we have about it, the better decisions we can make for its future. And we just don't really have a lot of information. So we're kind of flying with our eyes half shut. Some more far-reaching future work would be a microplastic study. This was one of my original intentions. In the stomachs, the gastrointestinal tracts of these fish, as well as some invertebrates that we've been collecting, are definitely microplastics. You've probably been hearing about this, that they get found in more and more places. Human bones was the most recent one that I found. So microplastics are obviously plastic on a very, very, very tiny scale. And for fish, these are a huge problem because they can get into their gills, particularly juvenile fish. And plastics are sharp, even at a microscopic scale. And this can lodge in their gills and it can do physical damage. It can block their ability to breathe. Plastics can also absorb toxins. They basically act as microscopic sponges that can then be inside the tissues or the stomachs of the fish. They can fill up the fish's stomachs. Maybe you've seen the sad photos of the albatross chicks at Midway Atoll. Think of that just smaller. So it would be interesting to compare, and this was my very original, like way back idea, compare the 1963 and the 1928 fishes and their microplastic load to the modern stuff, and do a time study, a longitudinal study, to also look at other Bay Area urban lakes. And then fascinatingly, Oakland has a sister city in Japan that has an urban lake that has been managed for roughly the same amount of time. And again, another big thank you to Andrew Alden for making a post about this. This is Ohori Park, and this was an estuary-fed lake and is now entirely freshwater. But it is weirdly similar to Lake Merritt. And I think that this would just be a fascinating sister study. So that is it for me with 5 minutes to spare. If you have questions or would like to volunteer or anything, I am always looking for people to go out with, go in the water. Please, please, please send me an email. This is my work email, and no emails will bounce from this. So I will stop the share, Katie. Do you want to take
Rebecca,
over the
this
share?
is amazing. You lower my hand. This is fascinating. You did such a great job. I know we're all going to want to talk to you about this, and thanks so much for encouraging people to come to Lake Merritt to really interesting um, questions to be pursued. I'm going to take a moment before 8 o'clock to say some thank yous. So, yeah, I want to thank everybody here for coming. Special thanks to Rebecca Abrams for pulling all of this amazing information and energy and excitement together. I want to thank our new voices, our rising externs and fellows, Team Oakland, Lake Merritt Institute, Achieve, the Boating Center, Save the Bay, the cadets, and all of Oakland for caring about our lake and wanting to know more about it. Coming up, yes, on Friday, November 7th, Lakeside Chat number 60. Damon Ty is going to return and talk about UV biofluorescent and bioluminescent organisms of California. This should be fascinating. He always does a beautiful job. And we'll be getting that Eventbrite out and more information soon to everybody. We want to encourage people to check out the Autumn Lights Festival, which is on at the Gardens of Lake Merritt from the 16th to the 18th of October. And I think Janai was going to put that link in the chat. The California Naturalist Certification course that we have launched starts tomorrow. I'm very excited about this. You'll hear more in future chats, and it'll be a lot of fun, and hope to learn a lot and bring a lot of people together. Our Adopt a Spot mornings in the marsh and water testing and plankton dates will be forthcoming on our Facebook and on our link tree as we get them organized. So wish us luck, and we're going to have a lot of important finds and things to share from the Urban Estuaries California Naturalist Certification course. I'd like to thank the City of Oakland KTOP Channel TV, Channel 10, for rebroadcasting all of our Lakeside chats. They will be rebroadcasting the show tonight after light editing. And the plan is that on Sundays from 6 to 7 p.m., they rebroadcast one of our programs. The first 2 Sundays are dedicated to older programs, and the last 2 Sundays are dedicated to the current one. If you enjoy what we do, please consider making a donation to Rotary Nature Center Friends. We are so happy for all of the help that we have received, allowing us to launch the California Naturalist course and to provide scholarships for many of the people who will participate and otherwise would not be able to take this course. Special thanks to our producer, Rob Lamone, for media and videos. I and David Wofford are the co-chairs of Rotary Nature Center Friends. Kirsten Furman, Betsy Schultz, Rebecca, Miriam, Rohan, Leander, Richard, and many RNCF volunteers have been involved in making possible our programs in person and our Lakeside Chat series. So we want to thank everybody for that. It's been such an amazing evening. Rebecca, you were able to stay a bit longer to take for some discussion.
Totally.
Awesome.
Yeah, let me share my screen again, and that'll have my email up on it just in case anybody
Fantastic.
has a grand desire to email. Always down for that.
Yeah.
And I see 3 questions in the chat that I can answer. I'm hoping the people are still here. The first one, I'm going to need clarification on this. It says, "What about oysters?" And it's from Mary. Mary, are you still here? I don't know.
Mary answered, what about the oysters? Curious about oyster populations
Oh,
over time, I believe.
interesting. Okay.
They clarified in chat.
Okay, so Katie, correct me if I'm wrong. I don't know if we have oysters in Lake Merritt.
Yeah. So there was a lot of interest in establishing oysters to filter the water and also to increase the native oyster populations. However, that project has not proceeded with much, much happening, and I think that there really currently are not oyster populations. There are a few oysters there was one that was pulled up by Damon off of a dock in 2021, but it's just like occasional, and it's not something that is a thing at Lake Merritt. We should go to, you can go to iNaturalist and search for the California native oyster there and see where it is distributed in San Francisco Bay based on iNaturalist observations, which, are need to be, verified, of course, once they're there. But no, so it has not been established as a big thing in Lake Merritt.
Yeah, we certainly find huge numbers of mussels. Mussels do filter water, but not nearly to the same extent as oysters. Um, there are uh, there is work going on to reestablish the oyster beds in San Francisco Bay, and that is starting to happen. Just a brief history note that maybe you don't need, but you will now. San San Francisco Bay used to be known for its oysters, and the beds were so large you could walk out into the middle of the bay on them. In fact, they were one of Mark Twain's favorite foods from Northern California. Um, but then obviously and this happened at the same time as the gold rush, oyster beds make a bay shallow. And when you dredge beds and then when you dredge the bottom of the bay and then you dump all of the nasty heavy metals from mining into it, gold panning mining, you make the water not so nice for oysters anymore. So they're, yeah, not so hot on the oysters right now. Sorry.
I'm going to drop into chat a search in iNaturalist for Lake Merritt and oysters of all types, and it's just not a big thing here. So I will share that in the chat. We also had a talk, a Lakeside chat, about oysters by Andrew Cohen. And that's an interesting story in itself. It was talking about different species of oysters that were found at Bay Farm and in other places that were actually not the Olympia oyster, but a different oyster. There was a lot of oyster growing in Lake Merritt in those times when that was a big thing.
That's really neat.
Oh, God.
During the 2022 fish kill, it appears the vast majority of the dead fish were forced into the lake as the estuary DO plunged and then the lake DO fell. So this is from Lee. Are you talking maybe about the bat rays? I'm just trying
we lost a lot of huge fish. I mean, there were, I don't remember the numbers, several tons, multiple tons of fish were removed, as well as the bat rays. And I don't think those were resident. I think they all wandered in during the oxygen shortage. And it felt like we were just the last refuge in this part of the bay.
Katie, what do you think about that? We definitely
Well, it's a great hypothesis, and really, it's a great hypothesis. We don't really have a good way to know it because we don't know where they came from, all those fish that were there. It seems clear that the algae heterosigma the offending harmful algal species came in from San Francisco Bay, from the south side of Alameda, and it invaded and probably pushed species ahead of it, and that's a very likely hypothesis, but we don't have data to really say one way or another. I am going to share a Lakeside chat by Andrew Cohen, and it is called The Mystery Oyster of San Francisco Bay. And it's one that was found, I believe, at Bay Farm. It's a very interesting story, and in the discussion, it ranged all over the place about, how big a thing were the Olympia oysters in San Francisco Bay, and some very interesting discussion on that. So I recommend taking a look at that.
And Lee, I think your question and your comment bring up a really interesting, a really important point about more and consistent water quality data. We can't really understand where a low dissolved oxygen event starts if we only have single monitoring stations or, a few people doing monitoring sporadically. We basically don't have enough coverage to get the information that could help us answer questions like yours. And so, again, part of what Rotary Nature Center Friends is working on is collecting as much data as possible, because I'll say it again, you can't really make informed decisions and policy about a place like Lake Merritt without data.
I'll concur. There was anecdotal information percolating in as the kill-off occurred, about people spotting fish die-offs around, up north along the edge, like say Berkeley. And it felt like it was coming down towards Lake Merritt. So that to me, that was just the other data point I had.
Yeah. Yeah. And I think that community data is very valuable. And again, we have to coalesce it somewhere. iNaturalist is not a bad place to do it. There are huge numbers of papers actually written from iNaturalist data. So if you're able to put your data somewhere, please.
I'd like to make a shout out to the San Francisco Estuary Institute. They're on the trail of this. There are many scientists who are gathering the kind of comprehensive data about water quality and plankton blooms and species and the dynamics of the water currents and movements and clarity and all that. It's a very fascinating and complex story that they're pursuing, and so it's and I think it's really wonderful that they're doing that. There will be I'm looking forward to going to the State of the Estuary Conference coming up at the end of this month, because there will be a lot of presentations related to trying to understand what happened and the dynamics of the living community and the biological community, climate change, the physical community, climate change, and so forth. It should be fascinating.
Okay, and then the last question that we have thus far comes from Deborah. Why is the algae harvested? And we got this question so many times as we were out there digging through the pile. The basic answer I can give you is aesthetics. Algae is, and this is macroalgae. This is meaning algae that you can see with your eyes and you could like scoop up with your hands. It is unsightly and sometimes it smells. And so once a year for at least a decade, possibly more, again, anecdotal reports, the city collects it, and because it's coming from an urban lake, in the case of Lake Merritt, it goes off to a city dump. Apparently other places where the water is a bit cleaner, it can be used as fertilizer. This year they were saying, my gosh, the people from the harvesting company were saying, my gosh, there's so little trash in it. And they were right. There was very little macro trash, which was kind of neat. But I think one of the interesting things that we've noticed just in the past, oh gosh, when is it? Maybe 3 months? Because that was in July, so August, September, October, yeah, 3 months, is right by the ramp, the boat ramp down into the water. There used to be a whole bunch of macroalgae there that lots of little killifish, the rainwater killies, Leucania parva, and the bay pipefish used to live in. And they got rid of all of it. And so the little fishies were gone. And just the past couple weeks, we've started to see them come back as that algae has come back. So yes, it's anecdotal, but the algae is habitat, not just for non-native species, but also for native species like the pipefish, which, and I didn't show a really good picture of them, I think are maybe our cutest fish in there. They look like a straightened out seahorse. The adults are a gorgeous emerald green, and they are hypersuction feeders. So they have a fused jaw into a tube and they drop down their mouth and they go and suck in their food. And they're just the tiniest of predators. And I think it's a real shame that we're doing something that probably removes their habitat, because their whole life is basically pretending to look like eelgrass and not really move anywhere. Yeah. And so, okay, and yeah, so to Carl's note, like which fish are being eaten by which birds, this is also something that can really come from that community data. and the photos that people take. Lila takes phenomenal bird photos, often with fish in the beaks of birds, saying, "What fish is this?" And that is super, super cool. Because we can see that they are kind of preferentially eating like the yellowfin gobies, which are big. They're up to, you can't see how big this is, 6 inches. They're eating chameleon gobies, but they're eating also the top smelt. And so you can see that the birds are eating these non-native species. So yes, they're non-natives, but they're acting as a food source for native birds. And so there's a whole different food web that's happening, which is kind of fascinating. And again, I can't keep saying it enough. We need more data. There's just not enough data about this really fascinating and dynamic ecosystem that so many people visit every day. And I don't know if I can convince you to fall in love with it. If you're not already in love with it, please come down and look at all the cute fish with us. Or, the sea squirts. We'll get you by squirting you with a sea squirt. That's always a good one.
These are great questions. I
That's a
would
great.
love to know what, how different species of fish use the lake, because they have different life cycles, different lifespans, different ways that they interact with, like, the macroalgae or the bottom.
Yeah. And then Jerry, do we do stomach contents of the fish? That is definitely going to be one of the next steps, for along with that microplastic study, looking at what they're eating, because we preserve our fish in 100% ethanol. That allows for the preservation of DNA. We don't formalin fix and then put into 70%. We go straight into 100. That's a choice that the Berkeley Fish Collection has chosen. So we can see, yes, we can see what they're eating. And so we can see, are they eating other little fishies? Are they eating little arthropods, amphipods, algae, slightly bigger macroplastic? What are they ingesting also? And so that is the amazing thing about stomach contents, is you can start to build a food web from one animal, which I think is just the coolest thing. And you can do it from historic collections too. So those ones from 1928, 1963, and that weirdo from 1996, who knows what he's doing there. We can start to see maybe changes in that food web over time. I'm excited.
That is very exciting. Let's see. Well,
Can I ask a question of
of course.
Rebecca? This is John Hopkirk. I'm a senior fish scientist, and I was at Berkeley years ago. And strange as it may seem, 62 years ago, I had a job similar to yours, only instead of working for UC Berkeley directly, I was working for the UC system on a scholarship to curate the UC system's collections of fishes at the academy. They were housed there, and there was an agreement between Carl Hubbs and Paul Needham, who was professor of fisheries and one of my major professors. But I was the first person actually to see to identify the yellowfin gobies, and I was on the initial note on their occurrence in California. But I'm not very good with the computer, and I missed the first half of your lecture this evening. And how many did you see? Any native freshwater fishes, or did you see the tule perch, my favorite fish?
No, I would say that is one of the things about Lake Merritt, is that we see saltwater fish. Some of the gobies are more, they can handle, they can handle more fresh water, and they can move up into the streams. But we're not seeing we're not seeing things come down freshwater.
One time I recorded an eel from the pier at Berkeley, a snake eel. Have you seen any true eels in Lake Merritt?
No, but my lab does collecting in the local area. We've done the Berkeley Marina, the Berkeley Aquatic Ponds, Albany Beach, Cerrito Creek. I would love to talk to you, and I believe we have your fish. And also, I was just at the Cal Academy talking with their new curator, John Fong, who replaced Dave Catania just this year.
Right.
And he wants to give us back all of those fish.
Oh,
Yes, he's like, oh, you can take them now, because our fish collection only started in 2019.
Yeah, that's why I worked at the academy rather than
Exactly.
at Berkeley.
Yeah.
amazing.
Let's see, there's one other question. One of the ideas I always have thought for Lake Merritt that Laney College should do in conjunction with Oakland or whoever, just Laney. But they could put in what the Forest Service did up at Tahoe on Taylor Creek, a stream profile chamber. In other words, some way of recording the movement of fishes back and forth from the bay into Lake Merritt.
That would be
And it would be an educational feature that would add to the science of the area of Lake Merritt and the bay. There's a lot. There should it'd be wonderful if you could have sampling over in the bay at various locations. I know there's quite a bit from Hunters Point. Earl Harold had the Chinese fish shrimp net fishery analyzed or surveyed. They were collecting young of marine fish, some of them rock fishes even. But anyway, it would be an interesting comparison to see what they found back that would have been in the 60s.
Yeah, that would be very interesting.
That would be very cool.
Well, anyway, I'll shut up now and.
Oh no.
Oh, please.
My dog is belligerent. We're both 91 years old.
You should get his phone number.
I would love to stay in contact with you. Do you happen to be local?
I live in Albany.
Lives down the street from me.
Oh,
Yeah.
I would love to
haven't seen Katie in 50 years, I think.
Yeah.
Or no, not that long ago, Katie. I'm sorry, only 10 years ago, wasn't it?
Oh, I might have been 20 or 30.
Yeah, I
Yeah.
would love to come talk to you, and that.
One thing I've of course talked to or gotten to know briefly the fossil fish scientist,
Yeah.
Dr. Jewin, and
Yes.
her graduate student Jennifer. But
Jen
I was in hopes that somebody would take some of those rotten fish and skeletonize them so that the university could have an element collection, a collection of isolated skeletal parts for archaeologists and for fish scientists.
I'm starting one right now. I have 2 fish processing in our domestic colony.
Oh, good for you.
But Jen Hofflich is just a lovely human being. Yes. Yeah.
Okay. Thanks.
Okay. Question from Jason Yang. How did fish communities in the mid-1800s differ from those we see today? Do we have any good ideas from historic information? That's the dream question. How do we find out? Maybe fishing records, historic photographs. We know that there are always those historic photos of like, look at the size of this halibut that I caught, and it's like the size of a human, and now we're catching smaller ones. Technically, we can look at like middens, kind of. if you want to go back even farther, we can look at things like shell mounds and middens, and that is where those osteology element collections come in for identifying fish. But a lot of those were purposefully destroyed, and also destroyed. Yeah, we know. I once talked to Jerry about Jerry Lips about doing a core sample of Lake Merritt to see like to look for like fish subfossils, and he's like, "What are you talking about? It's been dredged so many times." So that dashed my hopes. I think maybe our best bet is historic records. If anyone has other.
I'm also wondering, of course, this is very dreamy sort of imaginations with regards to the kind of modeling, capabilities we have today, as well as maybe projecting from similar estuary situations in other parts of the state, and trying to sort of recreate some scenarios. But as some kind of research in public education, or even musing, it would be really quite interesting to know, especially it may reveal like a vision for what we like to see different, here.
Yeah, I think maybe it's possible. I was just talking to our birds curator, and they were saying that they were using historic bird nests from the turn of the century to pull plant material to decide what to do replantings, native replantings. And I'm wondering, can you do stomach contents from, it's not common to preserve birds in ethanol, but you could, if anybody has those from like turn of the century birds, to see what fish they were eating and maybe go from that. Like it's a stretch, but maybe we can get lucky. Or it's more common with reptiles. So, we know that there are reptiles that eat fish with moderate regularity. So we could look at that. Funding. But yeah, I mean, I think that would just be absolutely fascinating. Like, again, how do we move forward with any sort of like conservation or restoration plan if we don't even know what point we're trying to restore to? Because we're just kind of being like, oh, well, it must have looked like this. It must have been this. And it's like, no, this place has been changed by humans since 1850. I mean, it is so heavily modified.
Right.
Yeah.
Geomorphologically, it has so changed and continue to be modified.
Yeah. Yeah, but that would be really cool. Yeah, how do we do that? Yeah, I tried doing it through a photo dive, and I really thought I would find more pictures of people fishing at Lake Merritt, and I did not. I went through all the Berkeley accessible photos through the UC archives, and people just didn't fish at Lake Merritt in that case. I haven't gone through Oakland's library or Berkeley Public Library yet, but
there may be some at the Lake Merritt Institute. I don't know. That might be a place to check.
Yeah. Yeah, I think
there. Yeah.
And I think, like you said, using AI software basically to identify something like that or start to ID photos to be hand checked later is a really good use of that. It's a kind of a time saver. Could be interesting.
Very.
Really appreciate it. Thank you. I really enjoyed your presentation.
So cool.
Thank you, Rebecca.
That's wonderful.
I'm going to talk about this forever.
So, yes, please do, communicate with Rebecca And consider coming to Lake Merritt and seeing what we get in our fish traps, and hopefully we'll have more information. Rebecca, I was wondering if you communicated with anybody at the women's college. Okay. At in Oakland. That there were some serious scientists doing collections there of mollusks in the 1850s. It seems probable that they would have somebody would have been collecting fish there. Probable. But what happened to that is another thing.
That's the question. Where did it all go?
Yeah.
It should be, but then. So yeah, some of it may just require like going there in person and just asking people to dig.
Yeah.
Well, that's fine.
Right. Well, this has just been amazing. Thank you so much. And thank you to everybody. Do we have more questions that we want to deal with? Or I'm sure Rebecca would be happy to take emailed questions. And, please do get in touch with us. One of the cool things about what Rebecca does is she reaches out to the community, and she's out there doing research and meeting people and talking about what she's learning. And that's really very cool. And something that Rotary Nature Center Friends aspires to facilitate and do. So really, really had a wonderful time this evening. Anyway, so I think it is time for us to close. If there are no more questions, let's all give Rebecca a round of applause. Yay. It's really great that everybody has been here tonight. It's such a wonderful day to have everybody here, to hear from all these voices. It's been very cool. Thank you all so much.